video number 6 for your IB Biology Topic 1, and this video is looking at membrane transport. So we're looking at Unit 1.4, Cell Membrane Transport. Okay, so basically what we're looking at is how substances, cross our phospholipid bilayer. Okay, so whether it's getting in and out of the actual cell or whether it is substances crossing in and out of organelles, how do different substances pass across that plasma membrane? And there's two categories, and we can see them in front of us here. We've got that passive transport, which doesn't require any energy for the particles of the substance to move across the membrane. Now, they can do this via diffusion or facilitated diffusion. We'll look at both of them shortly, but no energy is required for the substance particles to move from one side of the phospholipid membrane to the other. Okay, those molecules can pass across with no energy requirement. The second type of transport that we've got is what we call active transport, and this does require energy because the required molecules are moving against what we call the concentration gradient, and we will look at that again shortly. So passive transport requires no energy, active transport requires energy from ATP. Okay, so looking at this passive transport a bit more closely, we've got three different methods of passive transport. We've got simple diffusion, okay, and that's where molecules pass directly across the membrane. That's small enough to pass between the phospholipid molecules. We've got facilitated diffusion, and this involves some kind of channel that the molecules can pass through. The molecules are too big to pass between the phospholipid molecules, but they can get through a channel protein or something like that. And then we have osmosis. Now, osmosis is where water passes across a semi-permeable membrane, but it is against a concentration gradient. So it's just slightly different to simple diffusion. We'll look at each of these in a bit more depth. Okay, so the first thing to just get your head around is the whole idea of concentration gradient. Okay, so an area of high concentration has lots of particles of a particular substance. Okay, so lots of molecules of one substance, and an area of low concentration has a lesser amount of molecules of a certain substance. Okay, and our concentration gradient is from high to low. Okay, so in simple diffusion, all right, it's what we call that passive movement of particles, so it doesn't require energy, from a region of high solute concentration, so there's lots of particles of a lots of molecules of a substance, to a region of a low solute concentration, so an area that has a low number of those molecules. Okay, so it's trying to find an equilibrium. So the molecules are trying to balance up on either side of the phospholipid bilayer. So as we said, requires no energy. That's that passiveness. The movement occurs down that concentration gradient, so from a high concentration to the low concentration to form the equilibrium. Once that equilibrium is reached, there will be no net movements. There'll be no further movement of molecules. So that's our simple diffusion. Now, the next step on is this idea of our facilitated diffusion. Now, this occurs with the larger molecules. In our simple diffusion, those molecules are small. They can pass through that phospholipid bilayer. Okay, whereas facilitated diffusion, this is for those molecules that are bigger, or even some certain ions. Okay, it's still passing down our concentration gradient, and these larger molecules or ions pass through what we call protein channels, okay? And these are proteins that are embedded in our phospholipid bilayer, and they allow these molecules to pass through them as such. Okay, now before we move on to our third passive process, osmosis, we just need to understand a little bit about water potential. Okay, now an area with a higher concentration of water potential has more particles of water in it than an area with low water potential. So if you look at this diagram here, on the left, there are more water particles compared to solute particles. So the left-hand side has more water potential. On the right-hand side, there are more solute particles, solute to water particle ratio, basically. Okay, so that's a higher solute to water particle ratio there, so it has a lower concentration water potential. Okay, so what happens in osmosis is the water wants to reach an equilibrium. So it wants to literally dilute the solute, for example, on the right-hand side, so that the solute concentration is the same on both sides. Just to let you know, osmosis is really important in potato production. So here we have two diagrams, and you'll see that we have the semi-permeable membrane down the middle. Now, if we look at diagram A, you will see that on the right-hand side, there is a higher concentration of sugar. Okay, there's more red particles over there than there are on the left-hand side. So basically, what the water wants to do is make the concentration of the sugar the same on both sides. So even though it's moving against pressure, okay, the water will move into the right hand side. You see that pressure increase there, okay, to ensure that the concentration of the sugar is the same on both sides. Osmosis is a fascinating process. Okay, so again, it is a passive movement, so it requires no energy. This is just unique to water. Okay, and it's moving across that semi-permeable membrane from a region of a lower solute concentration to a region of higher solute concentration. Passive, no energy is required. It will occur until that equilibrium in terms of solute concentration is reached. So then there'll be no further net movement. Now, this is important in cells because this whole concept of osmolarity. Okay, now you may have heard of isotonic energy drinks. Okay, and if we have a look, what isotonic means is balancing, basically. All right, we've got three cells here, and they're in different solutions. Okay, they're in a hypertonic solution, an isotonic solution, and a hypotonic solution. Now you'll see in the hypertonic solution, water is moving from the cell out. So the solute concentration in the hypertonic solution is more than within the cell. So the water is moving out of the cell. If we look at the isotonic solution, we've got that equilibrium is balanced. Okay, so the movement in and out of the cell is balanced. And here with the hypotonic, the solute concentration within the cell is higher, so water is moving from the solution that the cell's in into the cell. You'll see it's actually the vacuole here, so we're looking at plant cells, actually that vacuole, potentially plant cells, that vacuole is actually where the water is held. Okay, and when the water has moved out the cell and the cell's starting to lose its turgidity, we say it's plasmarized. When we see here, when the cell is in its equilibrium, so at its best state, we say it's flaccid. And if that cell has too much water in it, we say it's turgid. So the concentration of a solution is expressed as the total number of solute particles per litre. A hypertonic solution is one with a higher concentration of solutes than another solution. Hypotonic has a lower concentration of solutes and another solution. And if the two are the same, we say they are isotonic. What does this do to a cell? Okay, if we have a high water potential outside the cell, okay, then that water from outside the cell wants to move into the cell. Okay, what happens if the cell takes on too much water? It will explode. If the water potential inside and outside of the cell is the same, then there's no movement of water, our cell remains the same. And if our water potential outside of the cell is less and it's higher inside, our water is going to move out and then our cell becomes plasmarized. Okay, so this is hypertonic, this is isotonic, and this is hypotonic. Turgid cells will explode. Plant cells takes a bit longer because they have that rigid cell wall, that cellulose cell wall. Okay, so in terms of this passive transport, kidney dialysis is a great example of this in practice. Okay, and if you have a look here, we've got so this patient, their kidneys have failed and they now need some help at filtering their blood. Okay, so you can see the blood comes from the patient into the dialysizer, and you'll see we've got this semi-permeable membrane here. Now the fresh dialate comes through here. Okay, and because there is none of the molecules that we're trying to remove in it, that diffusion occurs and all those waste substances move in and can be removed from the patient's blood, and the fresh blood goes back into the patient. So just seeing this is one of your utilizations to understand where diffusion gradients can be used. Okay, obviously continuously used within the cells of your body, but this is an artificial means of mimicking a process in your body. Okay, so we've looked at passive transport, those processes that do not require energy. Now let's have a look at active transport. So active transport does require energy from ATP to allow these molecules that we're looking at to move across our cell membrane. Now, these molecules are too big to diffuse across our phospholipid membrane, but also they're moving generally against the concentration gradient. Okay, so meaning they're moving from an area of low concentration to an area of high concentration. Therefore, diffusion would not occur even if they were smaller molecules. So they require energy from ATP, and you'll see that in the diagram here. Okay, we're using a phosphate from our ATP, and then our ATP becomes ADP. Okay, so these are integral proteins in our plasma membrane, and they act as carriers. Many are specific to particular molecules or ions. They have enzymes to enzyme receptors to make sure they're taking the right molecules across. Some examples are what we know as sodium potassium pumps, and you'll see here that these pumps are moving sodium from a high concentration to a low concentration, and then vice versa, potassium from the high concentration to the low concentration. This diagram shows you the steps of a sodium potassium pump. Okay, so part of 1.4 is to have this understanding of membrane transport on the axons of neurons. Now, we will look further at this in topic 6, okay, but it's just introducing the idea of how these different membrane transports are important in neurons. Okay, so if you had to compare and contrast active versus passive transport, you should now have some ideas on how the two processes are different. Take a moment to note them down into a table. Now we move on to the process of bulk transport. So this is where the cell membrane itself pinches off into what we call a vesicle to allow substances to move into the cell, or a vesicle fuses with the cell membrane to release waste substances. Okay, so this is enabled by the fluidity of that phospholipid membrane, and we said, you remember, that it is quite a fluid membrane that we have. It does require energy. Now, there's two types of this bulk transport: endocytosis and exocytosis. Let's look at each one of them separately. Okay, so if we look at endocytosis, all right, we've got three different types here that you can see. The first we've got is phagocytosis. Okay, and that's where the membrane is kind of moving up to make a little, moving up to make a vesicle to surround a solid particle. Then we've got our pinocytosis, okay, where there is already an indent in the membrane and our molecule moves into that and then it gets pinched off. And then we've got our receptor-mediated endocytosis, okay. So endocytosis is the bulk transport of substances into a cell. And it's where that membrane is pulled inwards or invaginates to form that kind of pit, which pinches off and forms what we call our vesicle. The vesicle then moves into the cytoplasm with whatever molecule is within that, so whatever is required within the cell. So we have our phagocytosis, and that's when solid matter is engulfed. For example, white blood cells engulfing bacteria to then destroy that bacteria within the cell. We've got our pinocytosis, where substances are engulfed in a full solution. So they take in the solution that's around them. And this is an example with products of digestion. Okay, and we'll have a look at receptor-mediated endocytosis next. So you'll see there how those enzyme receptors on the outside of the cell knew that that was the molecule it was looking for, because the receptors on the cell fitted the receptors on the molecule, and that's how they know it's the right molecule, and then endocytosis occurs. So now looking at exocytosis. So if endocytosis is taking material into the cell, exocytosis is releasing material out of the cell. So we have in the diagram here a secretory vesicle that's come from the Golgi apparatus, and that will move to the plasma membrane. It fuses with the plasma membrane and then releases what's inside of it, so those substances to secrete. So this is that bulk transport out of the cell. So that vesicle fuses with the cell membrane and then releases what is inside of it. Okay, so at the end of this video, we've moved through most of the areas in 1.4. That section on the sodium-potassium pumped reactive transport in the axons, we will look at that further in 6.5. Okay, and we will be doing our osmosis prac once you've watched this video. See you in the next video.